Hello everyone. Today, Tam and I are going to be explaining myelination in multiple sclerosis. The neuron is the basic functional unit of the nervous system. It allows for neural communication within the brain and throughout the body. Like all human cells, the neuron has an outer plasma membrane to separate the intracellular from the extracellular, and a nucleus, acting as a hub to guide the activities of the cell. However, the neuron is unique in many ways. Besides the fact that it conducts impulses, it has unique longevity, sometimes living over 100 years. It stops being able to reproduce after assuming its role in communication, and it has a uniquely high metabolic rate. The neuron contains three main regions: the dendrites, which receive impulses from various other neurons, the cell body, and the axon and terminal branches, which transmit the impulses to other cells. These impulses are the main method of communication with neurons. These impulses, known as action potentials, are passed through the receiving dendrites and the axon by the action of ion channels, mainly sodium channels for depolarization of the membrane potential, and potassium channels for repolarization of the membrane potential. When looking at the axons of some neurons, one may see stripes or bands along their length. This is the myelin sheath, and will be our focus for this presentation. The myelin sheath occurs for a more rapid transduction of signals across the axon. This is because of saltatory conduction. Rather than passing through the axon in a continuous fashion, an impulse can jump between the exposed space in the axon, between the myelin sheath, the nodes of Ranvier. A host of cells are involved in the myelination process, and many throw glial cells, Schwann cells, and others into one broad category. So what are the differences? Glial cells is a broad category for all the non-neuron cells in the nervous system. This includes Schwann cells, but also cells like astrocytes, cells that guide the formation of synaptic connections between neurons. The main two cells involved in the production of the myelin sheath are Schwann cells and oligodendrocytes. Schwann cells are found in the peripheral nervous system, while oligodendrocytes are found in the central nervous system. Each myelin sheath in the peripheral nervous system has a Schwann cell. Here's how the myelin sheath forms. The Schwann cells first surrounds a portion of the axon. Then, the cell begins tightly wrapping around the axon, forming successive layers. Eventually, the layers compress and form the myelin sheath, with the Schwann cell on the outside. The myelin sheath formed by the Schwann cells acts as an insulator for the conducting impulse. Ions cannot leak out of the myelinated regions. Sodium channels are concentrated in the nodes of Ranvier in between. In the central nervous system, oligodendrocytes rather produce the myelin sheath. As opposed to Schwann cells, each oligodendrocyte has multiple processes that each form a myelin sheath.
Created using Powtoon.